Assalamu alaikum everyone. I hope you're all doing well, inshallah. My name is Muhsin Ali. I am a second year medical student, and today, inshallah, we'll be discussing foundational immunity for your FND final that's coming up, inshallah, on Thursday. So, without further ado, of course, if you have any questions at any point, feel free to interrupt me. You can unmute if you'd like, or you can ask in the chat box. Let's begin. Bismillah. So these are the objectives that we'll be covering today. We're going to be covering the two lectures that you took with Dr. Abdul Jabbar on innate immunity and adaptive immunity, and we'll elaborate on them separately, inshallah. So let's begin. So your immune system, what is your immune system? Like any other system, your immune system is made up of cells, which make up tissues, which will make up organs. And these tissues and organs are going to help in defending against a disease. Now, how does it defend against a disease? It's by doing two things. It firstly needs to identify what is causing the disease, and then it needs to get rid of it by killing it. And your immune system is comprised of two components within itself. The first one being your innate immunity, and the second one being your adaptive immunity. And throughout this lecture, inshallah, we'll be getting into the specifics of the both of them. So let's start with the innate immunity. So your innate immunity is present at birth, and it is also a quick response with low specificity. Now, what this means, by the way, this is going to make a lot more sense, inshallah, when we talk about the adaptive immunity, where you'll be able to make this comparison between the innate and the adaptive. So your innate, as the name suggests, innate meaning it's there from the beginning, is there at birth, present at birth. It's a quick response with low specificity. What this means, what specificity means, is that if you have, for example, a pathogen which is going to cause a disease enter your body, it is going to be, your innate immunity is going to be the first thing to activate in order to get rid of that pathogen. Now, in comparison, when we look at the adaptive immunity, inshallah, you'll be able to understand what the specificity exactly is. And of course, your innate immunity does not have any memory compared to your adaptive. And the cells involved are your phagocytic cells. And I need you guys to remember that your phagocytic cells are your neutrophils, your macrophages, and dendritic cells. So these are the three phagocytic cells that are going to be involved in your innate immunity. And an example of your innate immunity would be your physical barriers, such as your skin, which is, of course, if you have a pathogen, is going to be blocked by the skin, or the epithelium that is lining your internal organs. Now, this picture over here is adapted from Dr. Abdul Jabbar's lecture, and it's just to show you that your innate immunity, once there is a microbe that enters the body, there are several defense mechanisms that are innate within the human being, so myself, for example, that are going to prevent this disease or the microbe from progressing. So like we said, epithelial barriers, your phagocytes, which are your neutrophils, your macrophages, and your dendritic cells, and then your complement. Now, Dr. Abdul Jabbar didn't really go into the details of your complement, but I guess all you should know about this is that it's a cascade of reactions similar to the ones in your coagulation cascade, where it's proteins that are eventually going to lead to the death of the microbe. So that's all you need to know. And over here in the timeline, you can see that your innate immunity sets in between 0 to 12 hours of the microbe infecting the body. Right. So now the question is, how does your immune system, how does your body tell whether or not something is whether a cell is a pathogen or it's going to cause disease, or whether it's your own body cells? So in other words, how is it going to recognize yourself from non-self? It's done by these these molecules called PAMPs. Now, your PAMP is a pattern-associated molecular pattern. And what I need you guys to know about these PAMPs is that it's present on your pathogen. So PAMP for pathogen. And an example of this PAMP is a DAMP, which is damage-associated molecular pattern. And this, like the name suggests, damage-associated, is going to be released once the cell is damaged. Now we have a pathogen entering the body, and it's going to release these PAMPs in order to tell your body that, hey, okay, I'm a bad guy. I need the cells from my body to come to this bad guy and get rid of it. And how does it do this? Your immune cells have something known as pattern recognition receptors, and these are what are going to bind to your PAMP and DAMP. So over here, if you look at the picture over here, you have your pathogen, you have the PAMPs and DAMPs being released, and your immune cells are the ones that are going to bind the PAMPs and the DAMPs by your pattern recognition receptors. Now, an example of your pattern recognition receptor is your toll-like receptor, your TLR, and like we said, it's going to bind to your PAMP, and what it's going to do once it binds that PAMP is it's going to induce phagocytosis. Now, what is phagocytosis? Phagocytosis is just simply the engulfing of a microbe by a phagocytic cell. And what were the phagocytic cells again? It was your neutrophils, your dendritic cells, and your macrophages. So please remember those three cells. So I want you guys to follow my cursor here. So you have a microbe, which is this one over here, and you have your cell, your phagocyte, which could be one of the three that we just said. So the first thing that's going to happen is that your microbe is going to be ingested. So we have the PAMP being recognized by the PRR. The PAMP is being recognized by the pattern recognition receptor, and it's going to now what it's going to do is the phagocyte is going to engulf this microbe into a little vesicle. This vesicle over here that has the microbe in it is known as your phagosome. Right? So the first step is the ingestion of the microbe. Then we have the formation of the phagosome, which is basically just this little vesicle, this vacuole that has the microbe inside of it. Now what we have here, we have a lysosome, and we know that lysosome is an organelle that has digestive enzymes. This lysosome is now going to fuse with the phagosome. So it's going to form a phagolysosome, the phago from phagosome and the lysosome from lysosome, right? So you have fusion of the lysosome with the phagosome forming your phagolysosome, which is this one right here. Now, what these digestive enzymes from your lysosome are going to do is they're going to degrade the microbe that is inside it. So this is exactly what the last step is. It's the killing or getting rid of the microbe by the lysosomal enzymes. So again, just to quickly recap this, you have the ingestion, then you have the formation of a phagosome, and then you have the fusion of the phagosome with the lysosome to form your phagolysosome, and then lastly, you have the killing or the digestion of the microbe by the lysosomal enzyme that has just fused with the phagosome. Okay, so for this slide, honestly, this is just a filler slide. The doctor mentioned it really quickly, so I thought I just included for completion's sake. Basically, all he's trying to tell you here is that there is a specific component of your blood that is known as your formed elements. These formed elements are made up of white blood cells, red blood cells, and platelets. What you need to know is that your red blood cells are the ones that are primarily making up your formed elements. It's around 99% of the formed elements. And then the remaining 1% is going to be your white blood cells and your platelets. What you need to know about this is that your white blood cells are your neutrophils, your lymphocytes, your monocytes, your eosinophils, and your basophils. Now, these percentages here are just there for you to understand the percentage, like the ratio of which one is the highest compared to the lowest. And what you will need to remember, how you can remember this is by a mnemonic: never let monkeys eat bananas. So the first letter of the never, which is N, would signify neutrophils. And then the let would be lymphocytes, monkeys would be monocytes, and so on, going from highest percentage in the blood to the lowest. So from neutrophils being the highest to basophils being the lowest. And this is honestly the one thing that would be the most important from this slide. Right. So now, more importantly, the cells of your innate immunity. So like we said, we have the phagocytic ones, which are your neutrophils, macrophages, and dendritic cells. Again, I'm repeating this a lot, so it really sticks with you because it's important to know that your phagocytic cells are these ones. And now specifically your neutrophils, they're also called polymorphonuclear cells. Now, if you break this word down, it's a big word, it looks scary, but break it down. So you have poly, meaning many, morpho, coming from the word morphology, which means the appearance under the histological slide, and nuclear, meaning nucleus. So what this is saying, it's a cell that looks like it has many nuclei, and this is exactly why we call it a polymorphonuclear cell. So your PMN, it has around 3 to 5 lobes. This is the nucleus right here. So this is your neutrophil, and it looks like it has around, so I would say this is one lobe, maybe this is a second, and maybe a third and a fourth. So 3 to 5 lobes that are inside this neutrophil. So this is the reason that we call it polymorphonuclear. And then the remaining 2, of course, like we said, macrophages and dendritic cells. Now we have phagocytic cells. That means we also have non-phagocytic cells, logically. So these non-phagocytic cells are 3 of them that you need to know. So the first one being your natural killer cells, the NK cells. Now, what you need to know about these cells, the natural killer cells, is that they have 2 enzymes that they're going to release. The first one being perforin, and this kind of comes from the word like perforating, which is like to make a hole or to punch a hole into. And this is exactly what this enzyme is actually going to do. It's going to make a hole or punch a hole inside the microbe, and therefore all the cellular contents that are within this microbe are going to be released from these so-called holes, and therefore the cell dies. Right? So perforin is for hole punching, eventually leads to the cell dying. The second enzyme that we have is granzyme. And what this does is simply the same thing. It induces apoptosis. And what apoptosis is simply just cell death. So both of these enzymes that are released by your natural killer cells are involved in killing your microbe or your pathogen. The second one that we have is eosinophils, and these are involved in parasitic infections as well as allergic reactions. And then lastly, you have your mast cells, which are going to release histamine. And I want you guys to keep this, by the way, in the back of your mind, the eosinophils being present during a parasitic infection as well as allergic reactions, because later on when we look at antibodies, there's going to be a little bit of a connection there that you're going to be that you're going to need to form. Right. So now we said your body, your immune cell is able to recognize that there is a pathogen in your body through these PAMPs and DAMPs, and the PRR is going to bind these PAMPs and DAMPs. But the question is, how does your cell, for example, let's take a neutrophil, for example, how does your neutrophil get from the blood to the site of the infection in the tissue where the microbe is? And it's by a process called cell migration. So just to break this down, you have a macrophage. We said your macrophage is a phagocytic cell. What this phagocytic cell does, it is going to induce phagocytosis. So let's imagine here we have an infection. There is a microbe, and this microbe was engulfed right now by the macrophage. What this macrophage is going to do now is going to release two very specific things. The first one being chemokines. Chemokines are just chemicals that are going to tell the neutrophil that, hey, I need you to come to this specific site of the tissue so that you can have the neutrophil come in and phagocytose the remaining microbes. The second thing is your cytokines. Your cytokines are interleukin-1 and tumor necrosis factor, which is your TNF-alpha. And what these cytokines do is they're going to express certain proteins on the endothelium. So I honestly forgot to orient you guys. So let me just orient you real quick. So within these red lines is your blood vessel. So this is your blood vessel, and everything that's inside here is the cells that are part of your blood. And outside over here is your tissue. So your macrophage in the tissue is going to release chemokines to tell the neutrophils, okay, I need you to come to this site. Your cytokines are going to express certain proteins. The first protein that it's going to express on the endothelial cell is selectin. Okay, so selectin is going to be expressed on the endothelial cell. And what is this selectin going to do? Is there is a ligand on the neutrophil, so the selectin ligand, the selectin L, which is going to bind to the selectin on the endothelium, and is going to keep doing this in order for it to roll across the membrane of the endothelium of the blood vessel, right? So after you have this rolling, you need the cell to stick to its place, which is adhesion. And then the protein that is going to be expressed, or the receptor that's going to be expressed on the endothelial cell for this, that is again induced by these cytokines, is integrin. So the first one we have is cytokines are going to express selectin, which is going to allow for rolling of the neutrophil. The second one that we have is integrin, which is going to allow for the neutrophil to adhere to the endothelium. So first, it's rolling around, so it's going here, and then it sticks to that place. So I hope this is clear. And then lastly, now we have it in the blood, we need to be able to take this neutrophil from the blood into the site of the tissue where the microbe is. And how do we do this? We do this by another third protein that is expressed on the endothelium by the same cytokine, which is your PCAM. So you have your PCAM expressed, which is going to bind to your neutrophil, and it's going to allow your neutrophil to go through the endothelial. And the way that the way it's able to do this is by formation of these little slits in between the endothelium. And this is, by the way, going to be is going to be of significance when we talk about inflammation. So do keep this in the back of your mind that there is a little slit that forms inside the endothelium, so that your neutrophil can go from the blood vessel into the tissue and therefore go to the microbe and do its action. So is that all clear? Okay, so we have a few questions. Let's just answer these real quick. So we have someone asking, our body don't make histamine, right? So no, it is, like we said, if you remember, we said that from your non-phagocytic cells, we have your mast cells, which are going to release the histamine. So your body does release histamine. And specifically, your mast cells are the ones that release histamine. I hope that answered the question. Can you summarize cytokines' role again, please? Yeah, that's what we're going to do right now, actually. Okay. So again, just to quickly summarize, in text, we have the following events that occur in order for the white blood cell, any white blood cell, it doesn't have to be a neutrophil specifically, but in this case, we're saying neutrophil, to go from the blood vessel into the site of infection. So the first thing that needs to happen is that the macrophage is going to secrete these cytokines. Cytokines are, the examples of cytokines are your tumor necrosis factor, this TNF, and interleukin-1. What these cytokines are going to do is they're going to express selectin, integrin, and PCAM on the endothelium. So you have your macrophage, it's going to release cytokine, it's going to result in the expression of your selectin, integrin, and PCAM on the endothelium, right? Now, what you need to do with all this information is you need to be able to associate which protein does which function in the cell migration. So what that means is that we said selectin is going to be expressed by the cytokine, right? And what is the function of the selectin? It's going to bind to the selectin ligand from the neutrophil in order to allow this neutrophil to roll across the endothelial cells, right? So you need to be able to associate rolling with selectin, adhesion with integrin, which is again another protein that is expressed by your cytokine. And then lastly, the migration through the endothelium, the slit that we formed before, in order for the cell to go, the neutrophil to go from the blood vessel into the tissue by your PCAM. So again, just to quickly summarize, rolling is by selectin, adhesion by integrin, and migration through the endothelium so it can go into the tissue by your PCAM. Is that clear? Okay, so we have a few more questions. There's someone asking, do we need to know the examples of cytokines, or are they not important now? Keep them in the back of your mind, because the doctor did mention them, that the specific cytokines that are going to express those proteins are your TNF alpha and your interleukin-1. So do remember them just for safety's sake. Someone else asked, do these adhesions slow it down or stop it completely? So I guess honestly, it would just, it would stop it. Because so imagine this, we have it rolling, so it's moving across. Now you need it to stop so it can go through. So we have these over here, right? We have the membrane. It's rolling, rolling. It stops. Now it's going to bind to this PCAM so it can go inside, into the tissue. Clear? I hope this is clear, because this is a little, heavy on the concept. Okay, alhamdulillah, all clear. All right, so now we have a pop quiz. I would like someone to, if they could, unmute their mic and read, and if not, then I can read the question, but I need you guys to answer, of course, in the chat. Do we have any volunteers? No volunteers? Okay, go ahead, Omar. If you could unmute your mic and read the question real quick.
3-year-old boy presents with recurrent skin and soft tissue infections and a delayed umbilical cord separation. Laboratory studies reveal marked neutrophilia, but neutrophils fail to migrate to sites of infections. Genetic testing shows a mutation affecting integrins. Which of the following neutrophil functions is directly impaired due to this defect?
So is the answer firm adhesion to vascular endothelium? Is it phagocytosis of bacteria, migration through the endothelium, or chemotaxis towards chemokines? Okay, so we have a few answers in the chat. A, that is the correct answer. Thank you, Amar, for reading the question. I appreciate it. So what do you need to know about this? There's a lot of text here saying about 3-year-old boy, skin infection, this, yada, yada. You don't need to know all of that. The question is right here. Genetic testing shows a mutation affecting integrins, right? So if you remember, what were we supposed to associate with integrins? It was the adhesion of the cell adhesion to the endothelium, right? So the answer is A. I hope that makes sense. So like we said, your integrin is involved in adhesion. So if it says that there's something affecting your integrins, that means that a function that would be impaired is the adhesion, which would normally be there with the proper function of the integrins. Right. I hope that's clear. Now, okay, so someone is asking, it would be C if it's PCAM. So let's look at the question real quick. So let's say it says genetic testing shows a mutation affecting PCAM. So it would be C, migration through the endothelium. Exactly. And again, just to make it stick, migration is through PCAM, adhesion by integrin, and rolling by selectin. All right. Now, the events that happen after your infection is that your cells are going to be released, of course, by the same process, the migration, and they're going to go into the tissue. So the first cell that is going to appear after there is an infection, right? After the microbe has entered your body or tissue specifically, is neutrophils. So your neutrophils appear first. They're followed by macrophages or monocytes. Can someone tell me, by the way, what the difference between monocytes and macrophages are? Okay, so okay, exactly. Monocytes in blood, macrophages in tissue. That's exactly what I was looking for. So your monocytes are the cells that are in the blood. Once they go into the tissue, they're going to be called macrophages. So the first cell, again, the first cell that we find is going to be your neutrophil. Then you're going to see your macrophages increase. And then lastly, you're going to find your lymphocytes to increase. Now, again, the timing over here is not necessary for you to remember. It's just there to show you that, okay, the sequence of events is neutrophils first, then macrophages, and then lastly, your lymphocytes. Right. So now we're going to be talking, I think this is the last part of the first lecture, is inflammation. Inflammation is simply a response to an infection or trauma, right? So you have a microbe coming into your body, and what it's going to do is going to, do its disease-causing effect, and is going to cause an infection or an inflammation. Now, what I need you guys to remember is that the two things that will happen after the inflammation or as a result of inflammation in your body are as follows. You have vasodilation, which is basically just your vessel becoming bigger, and increased vascular leakage. Now, these two are important if for you to understand the signs that we're going to get to in just a minute. You're going to understand now what signs are in inflammation, and these two events, vasodilation and increased vascular leakage, are going to lead to these signs. So we'll just get to that in a minute. Before that, the types of inflammation, you have acute inflammation, which of course is short duration, and the main cell that is present in acute inflammation is neutrophils. So again, I need you to be able to associate acute inflammation with increased number of neutrophils. And then your chronic, on the other hand, is longer in duration, and the main cells that are present in your chronic inflammation are macrophages and lymphocytes. Now for the signs of inflammation. So the first one is redness. Now, if you guys remember, we said that the two things that inflammation is going to cause is vasodilation and increased vascular leakage. Now, vasodilation, like we said, is just the increase in the diameter of the blood vessel. What that is going to do is going to enable more blood to flow to that site of infection. So more blood means there's going to be redness. For the same reason of vasodilation, there's going to be calor or heat. So if you have like an infection, you look at it, you'll see it's red. This is because of the vasodilation, more blood flow. The same reason, there's more warm blood flowing to that place. So it's going to be more warm, of course, because the blood is warm. The third sign is tumor or swelling, which is the edema that we saw over here, by the way. If any of you noticed, we first have edema and then these neutrophils. This edema is the swelling, right? And the swelling is caused by your increased vascular leakage. Now, what does this mean? Do you guys remember when we mentioned the slits that are forming inside the endothelium? When the neutrophils are going out, there's also going to be some fluid from the blood vessel that is going to leak out into the tissue, right? Because of these slits forming. So because of these slits, there is going to be the fluid that is going to be moving into the tissue, and therefore you have this edema or swelling. And then lastly, you have the dolor, which is pain and loss of function. And these are associated with other chemicals that are released from the body, as well as the nerves that it has to do in that specific place. But you don't need to know the specifics of it. So again, you have redness, heat, swelling, pain, and loss of function. So I hope this is all clear. Now, once you have acute inflammation, which we said is the short duration one, and increase in which type of cell? In neutrophils. Acute inflammation means neutrophils. Once we have this, there is several outcomes that can occur. The first one being complete resolution. What this means is that everything is going to go back to normal. The vasodilation is going to go back. The vessel is going to go from vasodilation back to normal. The slits are going to close because you don't need any cells moving into the tissue anymore, and the microbe is completely eliminated or killed. Okay. A second outcome could be pus formation, and this pus, I want you to imagine it like a pimple. When you have a pimple, what's inside it is pus, and what pus is basically just dead neutrophils and cells that are being accumulated in that specific pocket. In that specific pocket. The third outcome is fibrosis, and this happens in non-regenerative tissue. So, for example, if you have an infection in the heart tissue, your myocardial cells actually, they're not able to regenerate once they die. So once it is going to be damaged by the infection, it's going to form a fibrosis, which is just collagen deposits from your fibroblasts, I believe. You should know this from your histology, kind of try to connect it. There's going to be collagen deposits in your heart, and therefore there's going to be fibrosis of that tissue, which is like the hardening of the tissue. And then lastly, we can have progression to chronic inflammation. So if it goes from a short duration of acute inflammation to a longer duration, this would change from acute to chronic, and this chronic inflammation would eventually result in tissue destruction. I hope that is all clear. So this is a checkpoint. If you have any questions, go ahead and ask, because we're done with the first lecture of innate immunity. Okay, so someone is asking, what did the chemokines do in cell migration? So let's revisit the slides real quick. So you have your macrophage that is going to release your chemokines and cytokines. Your chemokines are the ones that are going to tell, they're chemicals that are going to tell the neutrophils and the white blood cells that, oh, hey, this is where the infection is. I need you to come to this place. So it's like it's like signals or chemical signals that are telling the cells to come to that specific part of the body where the infection is. So I hope that's clear. Someone asked, macrophage is when they are in the tissue. Okay, sorry, this is the answer. Can you repeat the edema part, please? Okay. So we're on this correct slide. So we said that when your cell is trying to migrate from the blood vessel to the tissue, the last step that happens is the migration through the endothelium. And the way that this is going to do is by making these slits in the endothelium. So here you see the continuous endothelium is going to make a little slit. And this is for the neutrophil to go from the blood vessel to the tissue. While this is happening, there's also the fluids in the blood that are going to leak out from this slit. And that is what is going to cause your edema. I hope that's clear. Okay, perfect. So that's it for your first lecture. Let's move on to your adaptive immunity. Now, this is going to make you're going to have to connect the information that you remember from your innate immunity in order to make that comparison. So, adaptive immunity, otherwise known as your acquired immunity, is the immunity that is there that is going to develop throughout your life. So it's going to adapt, and it's going to adapt to the conditions of the body, and it's going to therefore cause an immune response. Now, in comparison to your innate, this is a lot slower and it's high specificity. Now we're going to get into the specifics of what this means. High specificity means that there is a trigger. There is something that is going to tell your adaptive immunity that, okay, you need to act now. In your innate immunity, it was whatever, if there's a microbe, it's the first thing that's there. There's no specific trigger. It just happens and the immune response is going to happen. But as for your adaptive immunity, there needs to be a specific trigger that is going to say, it's going to poke at this adaptive immunity and the cells that are involved to tell it, okay, I need you to activate now. And we're going to discuss what this trigger is in just a minute. And of course, in comparison to your innate immunity, your adaptive immunity has memory. And the cells that are involved are your lymphocytes. So the cells involved in your innate immunity were which ones? They were the phagocytic cells, which are your neutrophils, macrophages, and dendritic cells. In your adaptive immunity, you have the lymphocytes. The lymphocytes are your B cells and your T cells. What you need to know about these cells is that they are both made in the bone marrow. The B cells and the T cells are made inside the bone marrow. But then the T cells are actually going to migrate to the thymus in order to mature. So your B cells, the reason they're called B and T cells is because of the place of maturation, by the way. So the B cells mature in the bone marrow and the T cells mature in the thymus. But both B and T cells, the progenitor cells that are going to eventually make these B and T cells, are made inside the bone marrow. And then the T cells are going to migrate to the thymus in order to mature. Right. Now, your adaptive immunity within itself has two parts. The first one being your humoral response, and the second one being your cell-mediated response. Again, this was adapted from from Dr. Abdul Jabbar's lecture. And again, it's just to show you the cells that are involved in your adaptive immunity, your B cells and your T cells, and the time duration compared to the other one, your innate, where it was 0 to 12 hours. This one is more of 1 to 5 days, where these cells are going to start to act. Okay, so we said that we have B cells and T cells. Let's specifically talk about B cells now. So your B cells have a receptor. What this receptor is going to do is it's going to bind to an antigen. What is an antigen? An antigen is a foreign molecule that a lymphocyte, which are your B and T cell, is going to respond to. So let's say you have a microbe now. It has an antigen on top of it. What it's going to do is the antigen is now going to bind to, so this is your antigen, this is your B cell receptor, it's going to bind to the antigen. And the specific part that the B cell receptor binds to is known as your epitome. So if you follow my cursor here, you have the B cell, you have your antigen in black, and the green thing right here is your epitome. So this is the specific part that the B cell receptor is going to bind to. Okay? and one B cell is specific to a single antigen. So this is how I want you to imagine this is by this same diagram over here. So you have B cell number one and antigen number one. B cell number one is only able to recognize antigen number one. And you can see that the circular shape of the receptor is only going to bind the circle of the antigen. So B cell receptor one is only, sorry, B cell receptor one is only able to recognize antigen number one. Now your B cell receptor 2, which is another B cell, is going to be able to recognize another one. So that means that one B cell is only able to recognize a single antigen or a single antigen from the microbe. So again, in this case, it would be a triangular. And again, the green dot over here is the epitome, which is the specific part of the antigen that is going to be recognized by the B cell receptor. And over here, you can see that it's triangular in shape. So if you were to take antigen 2, it would not be able to bind to the B cell receptor 1 because they are not complement to each other. I hope this is clear. Okay, now for your humoral and your cell-mediated response. These were the two types of responses that occur in your adaptive immunity. So the main cells, what I need you to be able, again, to, it's just association. It's like buzzwords. You hear one thing and you need to be able to associate it with one thing. So you have your humoral response. The main cells that are involved are your B cells. So I want you guys to focus with me again on the cursor. You have your antigen, and it's going to bind to the B cell receptor. What's going to happen now is this B cell is going to undergo rapid proliferation. And what this rapid proliferation is in, immunology terms, I guess, is clonal selection. So again, if you look at the word clonal, meaning clone, it's going to make multiple clones of this B cell that has been bound to the specific antigen, and it's going to proliferate these cells in order to make more of these B cells that will be able to recognize that same antigen. Now, once you have many of these B cells, they are going to become activated into cells known as your plasma cells. So again, the timeline is as follows. Antigen binds B cell receptor. B cell undergoes clonal selection. It becomes a plasma cell. This plasma cell is going to secrete antibodies and immunoglobulin. These are interchangeable words. And we're going to get again into the specifics of antibodies and immunoglobulin in the next slide. So I hope this is clear. As for your so this was your humoral response. Your cell-mediated response is going to be primarily done by your T cells, right? So again, follow my cursor. You have the antigen. It's going to bind to the T cell, right, by something known as your antigen-presenting cell. You don't need to know the details of this, but it's basically a cell that is going to present the antigen, as the name suggests, I guess. It's going to show the antigen to your T and B cells. So it's going to present it to the T cell. The T cell is going to undergo clonal selection as well, for the same reason that the B cell underwent clonal selection. It's going to secrete proteins, and these proteins are going to eventually kill the pathogen. So I hope this is clear. Do we have any questions? Someone said, can you repeat it again, please? Sure. So we said that your antibody, your adaptive immunity is in two parts. So you have your humoral response first, and then your cell-mediated response. Your humoral response is by your B cells. So again, associate humoral response with B cells. They're going to multiply and divide really quickly, rapidly, by the binding of the antigen to the B cell. And then we're going to have activation of this B cell into a plasma cell, which is then going to release your antibody. And we'll get into the specifics of the antibody in just a minute. So this was for your humoral. Then you have your cell-mediated response, which is your T cell. So cell-mediated response is done by your T cells. Your humoral response is done by your B cells. It's the same idea for cell-mediated. It's just the difference of the cells. Again, you have the antigen being presented by the antigen-presenting cell, your APC, to the T cell. The T cell is going to become activated in that case by the binding of the antigen. It's going to proliferate again rapidly in the same way by clonal selection that the B cells underwent. It's going to secrete proteins, and these proteins are going to kill or eventually lead to the damage of the pathogen. So I hope that's clear. Okay, just to clarify, B cells are made in the bone marrow but mature in the lymph nodes and spleen. Okay, so the B and T cells are both made in the bone marrow. And once it's made in the bone marrow, your T cells are going to go from the bone marrow to the thymus in order to mature in the thymus. Your lymph nodes and spleen are what are known as secondary lymphoid organs. Your bone marrow and thymus are primary. This is not part of the objectives of this lecture. It should be part of another lecture. I'm not too sure if you've taken it, but basically, what the idea is that you have your primary organs, your bone marrow and thymus, and then your secondary organs is where it goes to after it's finished from its maturation in the primary organ. So in the case of B cells, from the bone marrow to the spleen and lymph nodes, and in the case of T cells, from the bone marrow where it's made to the thymus to mature, and then eventually to the lymph nodes and spleen to perform its action. So T cells aren't made in the thymus. They are not made in the thymus. They are made in the bone marrow with B cells, but they mature. This is the key word here is mature, that they mature in the thymus.
Assalamu alaikum.
Wa alaikum assalam.
I have a question.
Go ahead.
If the T cells mature in the thymus,
Yes.
the thymus it deactivates when someone matures, right? When they grow older.
Yeah, when they go
So
older.
are there like limited T cells in the body? Like it can't synthesize new ones?
Okay. So the idea of this here is the reason that when you grow older, your immunity becomes weaker. It's because of the same thing that you just said. Your thymus is going to disappear, right? It's going to be getting rid of by your body, and therefore it's not able to have any T cell maturation, and so your immunity is going to be lowered. So yes, when you grow older, the reason that your immunity is weaker is because your thymus goes away. That's the idea.
Oh, okay. But do we lose our T cells or not?
Yeah, they will of course decrease in number because they can't mature anymore in the thymus because the thymus is not there to begin with, right?
Oh, okay.
Yeah.
I get it now. Thank you.
Okay, of course. Okay, I hope this is all clear. Any more questions? Okay, so someone is asking, so T cell, okay, what about the memory T cells? Are they also lost? If you're talking about when you grow older, yeah, the whole process of the T cell. So if you follow my cursor here, the helper T cell, memory T cell, and then also your cytotoxic T cell, which are the ones that are going to kill the pathogen eventually, they're all going to decrease because it's like a chain of reactions, I guess. So you don't have the thymus, no maturation of T cells. So anything that follows the process of anything that follows maturation of T cell is not going to occur because of the absence of the thymus. Okay, so let's continue. So we said that your B cells are going to be activated into plasma cells, which are going to release or secrete antibodies. Now, what are antibodies? Antibodies are proteins made by your immune cells that are going to bind the antigen, right? So now, in specific, so I want you guys to follow my cursor again. You have this right here. This is the structure of an antibody. Now, your antibody can either be found on the cell surface. It can either be found on the cell surface or it can be found circulating in the blood freely. Right? Now we have 5 main types of antibodies. You have firstly your IgM, which is a pentamer. So I want you guys to again look at this over here. Pentamer meaning 5. So you have 1, 2, 3, 4, and 5 individual antibodies that are going to come together into a pentamer that is your IgM. And this IgM is involved in your primary response, right? Now, the second one that you have is IgG. It is a monomer, so it's just a single one. It is involved in the secondary response. It crosses the placenta, and it's the highest in amount in the plasma. So your IgM pentamer. How can you remember this? The way I remember this, I don't know if this is going to help you, is the amount of zigzags. So you have a good amount of zigzags in the M, meaning it's the biggest one, so pentamer, right? And it's involved in your primary response. Now, the way you can remember this is that IgM is, you can remember it as Ig mother, the M for mother. So when you get, for example, let's say you get hurt, you get a boo-boo, yeah? And then the first one to come to you to help you is going to be your mother, right? So your IgM is there in your primary response. Your IgG, however, is going to be the G over here in IgG is grandmother. So she's going to be the one that comes after your mother, right? So the same boo-boo you have, your mother comes first. So your IgM is going to be your primary response. Then your Ig grandmother, your IgG, which is going to be involved in your secondary response. And what you need to know as well for IgG is that it crosses your placenta. And this again is going to make a little more sense once we get to the immunization. The third antibody that we have is IgA. Now, this is a dimer. Again, a dimer meaning two. So it's just like this formed over here. You have two antibodies that are going to bind to each other to form the dimer. These IgA antibodies are going to be found in your secretions, so your saliva and your tears, and their function is to prevent the entry of the pathogen. And this would make sense if you think of the location. You have to associate, you have to be able to recognize the location and how that location is specific to its function. So let's say the saliva, for example. If I'm going to eat food now, there's a few microbes in the food that I'm eating. Now the saliva over there is going to try and prevent the pathogens from in the food to enter into my system, right? So your IgA in the secretions to prevent the entry of the pathogen. Now your IgD over here is a monomer again. So just to quickly, or we'll get to that in a minute, your IgD is a monomer and it's associated with your B cell receptor. So you remember how we said your antibody can either be membrane bound or free in the blood. So IgM, IgG, IgA, and IgE are all free in the blood. Your IgD is actually membrane bound on your B cell receptor, which it makes your B cell receptor. Right? Your IgE again is a monomer and it's going to be active in parasitic infections as well as allergic reactions. Now, who can remind me again? I remember I mentioned this when we were talking in the beginning of innate immunity. What is the significance of this parasitic and allergic reactions? Can someone remind me? What were the cells? Eosinophils, exactly. Thank you guys so much. So they're associated with your eosinophils, which again are also involved in parasitic infections as well as allergic reactions. So again, just to summarize this real quick, antibody is a protein that is made by your immune cells that is going to bind the antigen. So the antigen is like the bad guy, is the villain. The antibody is the hero. The villain is of course going to come from the bad people. So the antigen comes from the microbe. The antibody, the hero, is going to come from the good cells, so the immune cells. Your IgM is a pentamer. Your IgA is a dimer, and the rest are monomers. So IgM, pentamer, IgA, dimer. And again, the way you can remember this is that the letter A stands on 2 feet. So it has 1 foot right here and another foot right here. So it's a dimer, right? Your M, again, many zigzags. So pentamer, the biggest one. IgM is involved in primary response. IgG is involved in secondary response. Remember, mother, grandmother. And then lastly, IgD is membrane bound and your B cell receptor. And then IgE is associated with parasitic infections as well as allergic reactions that were also associated with your eosinophils that are part of your non-phagocytic cells in the innate immunity. Is this all clear? Is IgA the one breast milk? Is IgA the one for the breast milk from the mother? So we'll get to that in just a second, actually. Or yeah, and the slide after this. So the immune response, what you need to know from this is as follows. You have the exposure to your antigen the first ever time to an antigen, right? What's going to happen now is you're going to have the release of antibodies over this specific amount of time. So look at how many days it's going to take around, let's say 2 weeks, right? To increase and then decrease. Now, can someone tell me which antibody is going to be released upon the first exposure to the antigen? Which one of the 5 antibodies that we talked about? IgM, exactly. So IgM is going to be there for the primary response because it's the first time that your body is seeing this antigen. Now, upon second exposure, so this is the first exposure, now you have another exposure of the same antigen. What's going to happen now is look at how much faster this increase in the antibodies are. You see over here, it took around 2 weeks, let's say. Over here, just a few days and it increases. This is why we said that your immune response upon secondary infection gets faster, which would be your adaptive immunity, right? So this kind of just summarizes the whole innate and adaptive, your primary and secondary response. So we said again, primary IgM. So this one would be faster. After second exposure, it's going to be faster, and it's going to be which type of antibody? Your IgG, right? And this red is just to show you a contrast of there's a new antigen that is being exposed over here. So it's going to start the same pathway over here because it's a new antigen. And then if there was antibody for this same red antigen made, again, there was a second exposure, it would follow the same curve over here where it goes up really quickly and then plateaus eventually. Right. Now, lastly, this is, I think, the last part of this lecture, inshallah, immunization. Immunization is the process of becoming protected from a disease, right? And we all know, we've all, you guys all know about vaccines and all these things. This is all considered immunization. Now, there's two types of immunization. You have active and passive. Active is where your own body is making the antibodies by itself. So your own body is making the antibodies to the pathogen. And an example of active would be active immunization are vaccines. Vaccines, they're either dead or weakened pathogens that are going to be put into your body. Now, what this is going to do is going to induce the same response that we said. You have the innate immunity, the cells coming, the macrophages, all of those cells that we talked about, and then eventually move to your adaptive immunity, T cells, B cells, memory cells. And what it's going to do is the B cells, again, are going to make the antibodies by itself, right? So you yourself are making the antibodies in an active immunity. Now, your passive immunity, on the other hand, is involved in the transfer of antibodies. And where can we see this? We can see this in mothers that are breastfeeding their infants, their children. So because the baby is not able, because it's still new to the systems are not fully developed yet, the antibodies are not, the B cells are not able to make their own antibodies in the infant. So the mother, the breast milk from the mother is going to pass the antibodies from the mother that the mother made into the body of the baby. And that's why we said that it's not made within the person itself. And again, an example of this would be breastfeeding. Now, someone mentioned before, is it IgA? I think that that is going to be found in the breast milk. It's actually the IgG that is going to be moving from the breast milk to the from the mother to the infant through the breast milk. And again, so crossing the placenta, and I guess you could associate this as well with breastfeeding because it passes the placenta, the other one passes from the mother to the child. So it's IgG actually that is going to pass from the mother to the infant. Right, now this is the last slide. This is just to bring everything together and compare our innate and our adaptive immunity. So the development of your innate immunity it's there at birth. Innate meaning it's there from the beginning. Your adaptive, it develops throughout life. The specificity, which was the trigger. The trigger for your innate, it doesn't need a trigger. So it's low in specificity. Your adaptive immunity is high in specificity because it needs a specific trigger in order for the adaptive immunity to start. The response time, again, in innate is slow. Your adaptive is fast, because of the fact that we have the memory cells already and we know what the pathogens are and the microbes or the antigen there. So it's able to make the antibodies a lot quicker. And again, memory, of course, innate is no, and there is memory in your adaptive. And the cells that are involved in your innate immunity are your phagocytic cells. Again, just to remind you, phagocytic cells, macrophages, dendritic cells, and neutrophils. And the cells involved in your adaptive immunity are your lymphocytes, which are your T and B cells that are eventually going to do their things of making antibodies, and the other one making the T cells, making the cytotoxic T cells, and eventually the proteins. So that is it for this lecture. Before we move on, is there any other questions that I could answer? Okay, so I think no questions. That's great, alhamdulillah. Thank you guys so much for attending and the interaction. Really appreciate it. Please scan this QR code for feedback. Do remember that this feedback report has a few questions that we're going to test your knowledge on this specific lecture. And yeah, I wish you guys the best of luck, inshallah. You guys are going to do great, inshallah, on your exam. And yeah, assalamu alaikum.